Okay, hello to all of you. Let's start discussing chapter number 8. Um, Let me go into the slideshow mode. Okay, in chapter number 8, we are going to discuss about the lipids and proteins which are there in the biological membranes. So, we have already discussed about proteins in the last 2 weeks. So, first of all, the first week was focused on proteins, and the second week we discussed about the enzymes, and the majority of the enzymes they are protein in nature. So, in chapter, I'm not going to because we have already discussed, and I'm going to focus more on the lipids. So, first of all, we are going to talk about different kinds of lipids. Um, First of all, we'll start with the introduction of lipids, then different kinds of lipids, and then we will go into the biological membranes or the plasma membrane, and we are going to see how these lipids and these proteins they are present in the membrane and what function they are doing. So first of all, let's talk about what lipids are. So lipids is basically they are a category of biomolecules. So we have already discussed proteins, which is one of the biomolecules. Other than the proteins, the other biomolecules includes lipids, nucleic acids, and the carbohydrates. So we'll be talking about all these biomolecules as we will move forward in our course. So lipids, they are macromolecules, but they're a little different from proteins and the carbohydrates and the nucleic acids, because proteins, nucleic acids, and the carbohydrates they are polymers, which means polymers are those big macromolecules which are made of repetitive subunits which are called monomers. So lipids are a little different from the other biomolecules, that they are not polymers, but they are made of um, some of the components, but they are not made of repetitive subunits. So they are basically a class of organic compounds, and they basically includes fats and oils. We're going to talk about all these, and they are usually insoluble in water, and they are amphipathic in nature. So what does amphipathic word means? Amphipathic word means something which is dual in nature. So these lipids they have polar end as well as they have non-polar tails. So we can say that amphipathic molecule is that molecule that has one end with the polar water soluble groups, and the other end is non-polar hydrocarbon group which is insoluble in water. So they have dual nature. They have both polar groups which make them soluble in water, and they have non-polar groups which make them insoluble in water. So this is the classification of lipids. Uh, we can classify lipids into two broad categories. They can be open chain compounds or they can be fused ring compounds. See, so in the open chain compounds includes fatty acids, triacyl glycerols, sphingolipids, phosphoacyl glycerols, and glycolipids. So we're going to discuss all these different kinds of lipids. And in the fused ring category includes cholesterol, steroid hormones, and bile acids. So first of all, let's talk about what fatty acids are. Now, fatty acids is basically a long chain of carbons and hydrogens. At the end, they have a functional group called carboxyl group, which is a COOH group. So this thing we discussed in the last in the previous chapters also. Specifically, when we are talking about the proteins, we talked about the proteins are made of monomers called amino acids. Now, why they're called amino acids? Because they have a functional group, amino group, as well as they have another functional group called carboxyl group. Now, carboxyl group is acidic in nature because it can lose a proton. So anything which is a donor of proton is called an acid. So COOH group, carboxyl group, is acidic in nature. So that is why wherever this group is present, um, uh, like in the amino acids this group was present, that's why amino acids are called amino acids, because they have this functional group in them. Similarly, fatty acids are called fatty acids because it's a long chain of carbons and hydrogens, and at the end they have this carboxyl group which is acidic in nature. Because of the presence of this carboxyl group, these fatty acids they got the name fatty acids. That's why they contain this word called acid in them because of the presence of the carboxyl group. So they are unbranched chain carboxyl acids. They are usually 12 to 20 carbons long. Oh, I'm sorry. So they can be of two kinds. They can be, they're also amphipathic, which means they have polar groups as well as they have the non-polar groups in them. Now they can be classified into two categories. They can be saturated fatty acids or they can be unsaturated fatty acids. Now, saturated fatty acids are those fatty acids in which all the carbons they are linked to each other by single bonds. And unsaturated fatty acids are those fatty acids in which these carbons are linked to each other by double bonds. So here are some examples. Palmitic acid. This is an example of a saturated fatty acid. So in this, all the carbons, they're linked to each other by single bonds, and you can see how this structure is. And this is stearic acid. Here also all the carbons, they're linked to each other by single bonds. This is oleic acid. Here you can see there is a double bond. So because of the presence of the double bond, you can see how the structure of this fatty acid is. So there is a bend in the structure. So these two saturated fatty acids, which include palmitic acid and stearic acid, because all the carbons, they are linked to each other by single bonds, you can see how they have a straight structures. But here in the unsaturated fatty acids, there is a double bond, and because of that there is a bend, or we call it a kink in the structure. And here are a few more unsaturated fatty acids. This is a linoleic fatty acid. There are two double bonds, and you can see how the structure is bent. And here are some more examples of the unsaturated fatty acids. And this is again a table showing some of the examples of saturated fatty acids. This is lauric, myristic, palmitic, stearic. So and here they are given how many carbon atoms they have. Like palmitic acid has 16 carbons, stearic acid has around 18 carbons. And these are the formulas. And here are some of the examples of unsaturated fatty acids. So here is oleic acid, this is linoleic acid, then this is palmitic oleic acid, and this tells us how many carbons they have. Like oleic acid has 18 carbons, linoleic acid also has 18 carbons, and this tells us basically oleic acid has 18 carbons and it has one double bond. Linoleic acid also has 18 carbons, it has two double bonds, and this symbol shows us where is the presence of the double bond. So like this symbol refers to a double bond at the 9th carbon atom from the carboxyl end of the molecule. So this tells us the position of the double bonds. Now in the unsaturated fatty acids, as I said, there are double bonds. Now around the double bonds, the functional groups can be arranged in two kinds. If the functional groups are on the opposite side, then those kind of unsaturated fatty acids, they are called cis isomers. Okay, I hope you know what isomers are. Isomers are those compounds which have the same molecular formula, but they have different arrangement. They have a different covalent arrangement of the atoms. So as what do I mean by they have the same molecular formula? Which means that they have same number of carbon atoms or same number of hydrogen atoms. But how these atoms they are arranged, the covalent arrangement of atoms is different. So these unsaturated fatty acids, because of the presence of the double bonds in them, they can exist in the form of isomers, and these isomers can be cis isomers or they can be trans isomers. So cis isomers are those isomers which functional groups they are on the opposite side of the double bond, and trans isomers are those isomers in which the functional groups are on the same side of the double bonds. So the cis double bond puts a kink in the long chain hydrocarbon tail. Okay, so this is the nature of the cis isomer, that it puts a bend in the structure of the fatty acid. But if the fatty acid is in the form of a trans isomer, uh, the basically because the functional groups are present on the same side of the double bond, the there is less bend in the structure. Because of that, there is less bend in the structure, and shape of a trans fatty acid is like that of a saturated fatty acid. Okay, it's all because the trans isomer has the functional groups on the same side, so there is less bend, and the nature of the trans fatty acid is just like a saturated fatty acid. And greater the degree of unsaturation, lower the melting point. So basically, what does that mean? It means that um, if there if there basically if there is more unsaturation, which means if there is more double bonds in the structure of fatty acid, these fatty acids, stay at a they stay as a liquid at the room temperature. Now I can show you here. Like you can see that these are the unsaturated fatty acids. This is unsaturated fatty acid. This is unsaturated fatty acid. So these unsaturated fatty acids, they have a bend in their structure. So these kind of fatty acids at the room temperature, they stay in a liquid form. So on the other hand, the saturated fatty acids, because they are straight chain, so these kind of fatty acids, they can actually pack very tightly over each other, and these kind of fatty acids, they can exist as solid in the at the room temperature. Okay, so that is why more the degree of unsaturation, lower is the melting point, because more double bonds means more unsaturation, more bends in the structure, and they can stay in the liquid form as compared to their saturated counterparts. So they have lower melting temperatures or more melting points, because they stay more as the liquids. I hope it's clear. Okay, this basically just shows us this is basically, you have seen in this table that this is called fatty acid notations. So what does this mean? 16 means how many carbons are there, 1 means how many double bonds are there. So 18 carbons, one double bond, 18 carbons, two double bonds, 18 carbons, three double bonds. So this is called fatty acid notation. So it indicates the number of carbon atoms and the number of double bonds which are present in them. So 0 denotes 18 carbons and no double bond. So this is a saturated fatty acid. 1 denotes 18 carbons with one double bond. So this is a unsaturated fatty acid. So these were fatty acids. Okay. Now these fatty acids, so they are presence in different kinds of fats. So now we are going to talk about fat. So there's a difference between fatty acid and fat molecules. So fatty acid, as we just discussed, they are made of long chain of carbons and hydrogens, and they have a carboxyl group at the end. That's a fatty acid. Now what is a fat? Okay, fats are also called triglycerides, or they also called triacyl glycerol. Now what a fat molecule is? A fat molecule is made of glycerol, okay, and three fatty acids are attached to them. That makes a fat molecule. Now I can show you here. This is glycerol. Okay, now what is glycerol? Glycerol has three carbons in it. You can see these are three carbons, carbon number 1, 2 and 3, and of course carbons are attached with hydrogens, and at every carbon is attached a hydroxyl group. So in one glycerol there are three hydroxyl groups. So hydroxyl groups are functional groups. And so this is the structure of a glycerol. And at every hydroxyl group is attached a fatty acid, which we just talked about, the fatty acids. So this total makes a triglyceride or a triacyl glycerol, or we can also call it a fat molecule. So this is what a fat molecule is. A glycerol which has 3 hydroxyl groups. These hydroxyl groups are attached with 3 fatty acids. That is why it's called a triglyceride. 3 fatty acids attached to one glycerol. And a triglyceride is a fat, and the adipose tissue which is present in our body, that adipose tissue is, this is basically showing the picture of a triglyceride. Now in the triglyceride there is one glycerol with 3 fatty acids. Now these 3 fatty acids can be same, they can be different. Okay, depending on the basis of the kind of fatty acids which are present in the fat molecule, um, if the fatty acids which are attached to the glycerol are saturated, the fat is called a saturated fat. Example, butter. Butter is a fat. It is a saturated fat. So here you can see this is tristearine. So this is one glycerol to which 3 stearic acids they are attached. Okay, so you can see here they are in these 3 fatty acids, all the carbons they are linked to each other by single bonds. So this is a saturated fat. Now here you can see this is one glycerol which is attached to 3 fatty acids. So this is myristic, this is stearic, and this is uh, palmitoleic acid. And in this you can see there is a double bond, there is a bend in the structure. So because of that this is a mixed triacyl glycerol. So it has one of the unsaturated fatty acid in it. Now if the fatty acids which are attached to the glycerol are unsaturated fatty acids, then that fat is a unsaturated fat. Example, all the oils, olive oil, sunflower oil, vegetable oil, all the oils, they are unsaturated fats. Now the butter, which is a saturated fat, is solid at room temperature. Reason, because all the fatty acids which are there in the saturated fat of the butter, they are all saturated, they are all linked to each other by single bonds. So these fatty acid chains, they can easily stack on each other, and butter exists as a solid at room temperature. But on the other hand, in the unsaturated fats which has unsaturated fatty acids in them, and in the unsaturated fatty acids there are double bonds, and because of the presence of the double bonds there is a bend in the structure, or there is a kink in the structure. These fats, which we call unsaturated fats, they exist as oil, they exist as, not the oil, they exist as liquid at the room temperature. So that is why they have lower melting temperatures. Okay, I hope you understand that what triglycerides or so triglyceride, triacyl glycerol, or the fat word means the same thing. So they are accumulated in adipose tissues and they means storing fatty acids. So this is what the triglycerides are. Now the next category I'm going to discuss with you are called phospholipids. Okay, phospholipids, they are also called phosphatidic acids. Now what is the difference between triglycerides and phospholipids or phosphatidic acids is, just like triglycerides, they also have glycerol in them. Instead of 3 fatty acids, they have 2 fatty acids in them, and instead of the third fatty acid, they have a phosphoric acid in them, or a phosphate group in them. Okay, so phospholipids or phosphatidic acid, it's a compound in which 2 fatty acids and phosphoric acid are esterified to the 3 hydroxyl groups of the glycerol. So this is your glycerol. So here is first fatty acid, this is second fatty acid, and instead of third fatty acid, there is this phosphoric acid is attached here. So these are what are phospholipids, and these are very important category of the lipids, because phospholipids are present in the plasma membrane or the biological membranes, which we are going to discuss very soon. And the third category of the lipids is the phosphatidyl esters. Now, phosphatidyl esters, they are also called phosphoacyl glycerols. Okay, they're called phosphoacyl glycerols. Now, how they're different from the phospholipids? Basically, they have the glycerol in them. So here is first fatty acid, so here is second fatty acid, and at the third position, basically, the phosphate group is actually attached to an second alcoholic group. So they are classed as phosphoacyl glycerols. So the classification depends on the nature of the second alcohol that is esterified to the phosphoric acid. So here is a phosphoric acid. To this phosphoric acid is attached the second alcoholic group. Okay, so their structure is they have long non-polar hydrophobic tails. Because of all fatty acids, they have long non-polar hydrophobic tails, and they also have polar hydrophilic head groups because of the presence of these phosphates and the alcoholic groups which are attached to them. So they are also amphipathic in nature. And here are some of the examples of phosphoacyl glycerols. First is phosphatidyl choline. So these are the two fatty acid chains which are attached, and this is the phosphate group or the phosphoric group, and to this is attached this choline group. Okay, so that is why this is called phosphatidyl choline. And here are a few more examples. This is phosphatidyl ethanol amine. So it has basically same thing as the two fatty acid chains. What is the difference? To the phosphate group is attached the ethanol amine group. So that is why this is category of lipid is called phosphatidyl ethanol amine. This is phosphatidyl serine. So phosphate group is attached to serine, and there are some more examples here. So their polar head is charged because of the phosphate group, which is negatively charged, and then they have positively charged amino groups. The positively charged amino group is contributed by the amino alcohol, which is esterified to the phosphoric acid, which we have been all talking about. Next category is sphingolipids. Now, sphingolipids are different from the all the lipids we have just discussed because they do not have glycerol in them. So what do they have? They have, instead of glycerol, they have sphingosine in them. Now what is sphingosine? This is the structure of the sphingosine. So you can see it has also 3 carbons, and to these 3 carbons, this is the group which is attached to this carbon, and this so this is called and these sphingosines are basically long chain amino alcohols. Okay, so they are amino alcohols, which means they have an amino group. This is the amino group here, and they also have the alcoholic group. This is the alcoholic group. Okay, so this is the structure of sphingosine. So this is carbons. These are the carbons. They have 3 carbons, and to this carbon is attached this group, and there is double bond also here, and this is an amino alcohol. So to the second carbon is attached amino group, and to the third carbon is attached the alcoholic group. This is the structure of a sphingosine. Now, then there is another lipid called ceramides. Now, ceramide is basically sphingosine, and here in the sphingosine, you saw that at the second carbon there is an amino group. So to this amino group is attached a fatty acid. So to sphingosine at the second amino here at this carbon, which is to this carbon to which the amino group is attached, if fatty acid is attached here, it becomes a ceramide. Okay, and ceramide is, we are going to discuss the next category of lipids, which are called glycolipids, and this basically forms as a basic structure for the formation of the glycolipids. And this is sphingomyelin. This is a kind of a lipid which is present in our nervous system. So this is basically sphingosine. So the basic structure is the sphingosine, because this is a sphingolipid. Here is attached the fatty acid, and here um, at the alcoholic group, which is in the sphingosine, here is the alcoholic group. To this alcoholic group is attached the phosphate group, and to this phosphate group is attached the choline. Okay, so that is the structure of sphingomyelin, which is a kind of a lipid which is present in the nervous system. So sphingomyelin, this is a primary alcohol of sphingosine. Okay, so sorry, sphingomyelin is formed when the primary alcohol, which is this, okay, is esterified to the esterified to the phosphoric acid, and that phosphoric acid is attached to the choline. So it is basically sphingosine, and the here is the alcoholic group in the sphingosine, and this is attached to phosphate group, and this phosphate group is attached to the choline. That is what a sphingomyelin is. Now the next category of lipids is called the glycolipids. Now what are glycolipids? So glycolipids are those lipids to which sugars are attached. Okay, and ceramides, which we just saw here, ceramide which is basically a sphingosine, ceramide which is basically a sphingosine, and to this amino group, so in the ceramide is basically sphingosine. At the amino group is attached the fatty acid, and this forms as a parent compound for the glycolipids. So this is glucocerebroside. Okay. So what are cerebrosides? So cerebrosides are those glycolipids in which the alcoholic group which is present in the ceramide. So this is the alcoholic group in the ceramide. This alcoholic group is linked or bonded with the sugar. Okay, so glycosidic bond. So this is basically the ceramide which we just saw. So this is sphingosine, and this carbon which is attached to the amino group is attached to a fatty acid here, and this is the carbon which is attached to the hydroxyl group, and this carbon is on this carbon where the hydroxyl group is there, the sugar is attached. Okay, so glycosidic bond is formed between the primary alcohol group of the ceramide and a sugar residue, which can be glucose or galactose. So if we go back here, you can see this is ceramide. Okay, this is ceramide, which is basically sphingosine with this amino group, and to amino group is attached the fatty acid, and here is the hydroxyl group. So at this point is attached the sugar, and that becomes a cerebroside. Now because in this the sugar which is attached is glucose, so this is called glucocerebroside. Now the other categories of glycolipids, they are called gangliosides, and they are basically the glycolipids, and they have some complex carbohydrate moieties attached to them. Usually they have like more than 3 sugars attached to them, and one of the sugar can be sialic acid, and sialic acid is negatively charged, and it gives the charge to these gangliosides or to the glycolipids wherever this sialic acid is present. So this is example of some of the gangliosides. So you can see this is the basic structure, and this is showing that this is the GM1 ganglioside. So all these sugars are present in the GM1, and all these sugars, they are present in the ganglioside GM2, and these sugars, which is galactose and glucose, they are present in this galactoside. So these are all different kinds of sugars. This is galactose, this is N-acetyl galactosamine, this is galactose, this is glucose. So this is just showing that these different gangliosides, which are different glycolipids, they have different kinds of sugars in them. Now the next category of the lipids is called steroids. Now steroids, they are basically, their characteristic is that they are made of fused ring structures. So there are in the steroids, which is a category of lipids, uh, they can have rings like A, B, and C. So these rings, A, B, and C rings, they have um, six members in them. So these are six-membered rings, and D ring is a five. So this is A, B, and C. So they have six members in them. B also has six members in them. C also has six members in them. Members means the atoms. And the ring D has five members in them. Okay. So these steroids, they are made of these ring structures, and these ring structures, they are fused to each other. And under the category of steroids includes the sex hormones and the cholesterol. Now cholesterol is a very important steroid because this is present in our plasma membrane. So these are some of the structures of some of the steroids, like cholesterol. This is testosterone, this is estradiol, this is progesterone. Testosterone is a male hormone, estradiol is estrogen, this is a female hormone. So these are all steroids. Okay, so now we have discussed all the different kinds of lipids, and we from our previous lectures we already know the structure of proteins. Now we are ready to learn about the membranes. Now what are biological membranes? So biological membranes are the membranes which basically enclose the cells. So it's the outer boundary of the cells. So they cover the cell, and it is the boundary which separates the cell from the surrounding. So things can move from outside to inside to the cell through this membrane, and a lot of waste material can go from inside to outside of the cell through the membrane. And there are various organelles inside the cells. They are also enclosed by these membranes. So what is the basic structure of a membrane? So basically these membranes, they have in them lipids, and they also have proteins in them. And these proteins and these lipids, they are all connected to each other by non-covalent bonds. Okay, to understand the structure of a membrane, let me actually go to the last slide of this PowerPoint. Okay, so this is basically the structure of a membrane. Okay, so we call it a fluid mosaic model of the membrane. So I'm going to discuss with you soon why it's called a fluid mosaic. So this picture shows you the structure of a plasma membrane, or any membrane which is surrounding the different organelles. So you can see that these are the phospholipids which we just discussed. Okay, these are phospholipids which are the main ingredients of the plasma membrane. And also we just discussed the steroids. So among the steroids, we discussed various kinds of steroids. So one of the steroids is cholesterol. Cholesterol is present in the plasma membrane of the animal cells. So all this you see, these are four fused rings. This is all steroid. Okay, so first component which make the plasma membrane is phospholipids. Okay, phospholipid is that lipid in which there is a glycerol, and to glycerol there are two fatty acids attached, and instead of the third fatty acid there is a phosphate group. Those are what the phospholipids are. And we have discussed that these phospholipids as well as the cholesterol, they are all amphipathic in nature, which means they have polar groups as well as they have the non-polar groups. And the third important component of a plasma membrane is the proteins. So these are all different, these blue structures you see, these are all proteins. And the different kinds of proteins which are present, which I'm going to discuss soon. So the third component of plasma membrane is proteins. And also there are some sugar chains attached to the lipids as well as to the proteins. If the sugar chains are attached to the lipids, they are called glycolipids, or if the sugar chains, they are attached to the proteins, they are called glycoproteins. So this is the basic structure of a membrane. So this is cytosol, which means this is inside the cell, and this is outside. This is all outside the cell. So this is the plasma membrane which is separating the cell from its external environment, and things are coming from outside to inside through this plasma membrane. And this plasma membrane is made of lipids as well as proteins. So why it's called fluid mosaic model? Mosaic, it's called mosaic because it's a mixture. It's a mixture of lipids as well as proteins, and also there are sugar chains. Why it's called fluid? Because all these lipids and proteins, as I just said, they are actually linked with each other by very weak bonds, non-covalent bonds, by hydrophobic interactions. They are connected to each other, and there is actually a lateral movement of these lipids as well as proteins. They are actually laterally moving, and this membrane actually maintains a fluid nature. Okay, and the fluid nature of the membrane is very important for the functioning of this membrane, because this membrane is a very dynamic entity. There's a lot of transport taking place across this membrane. The cell is living only because of the transport taking place across this membrane, and this transport which is taking place across the membrane is all because of the lateral movement of these proteins and the lipids. Because of the lateral movement of these proteins and lipids, the membrane is able to maintain its fluid nature. So that is why this model is called fluid mosaic model. Fluid because the membrane is like a fluid. Mosaic because it's a mixture. It's a mixture of lipids and the proteins. So now let's go and talk in more details about the structure of the membrane. So I just gave you some introduction about the membrane. Okay, so this basically talks about various things about the membrane, which I've already explained. And the major force driving the formation of lipid bilayers, I'm going to talk about that, is hydrophobic interaction. So as I just said, the main component of the plasma membrane is the phospholipids. Now these phospholipids, this is the structure of the phospholipids. This is the polar head, these are the non-polar tails. Okay, now so this is the polar head, this is the non-polar tail. So polar heads, because they are polar, they are soluble in water, and this side of the phospholipid, this is all non-polar or hydrophobic, which means this side of the lipid is insoluble in water, or it's hydrophobic, which means this is afraid of water. So because this phospholipids, they are making our plasma membrane. So let's go back to the model which we just discussed. We can just see here. So this is outside the cell, this is inside the cell. Outside the cell there is a water, inside the cell there is a water. Okay, so there is a water outside, there is a water inside. So how these phospholipids are going to align themselves? So this is the structure of a phospholipid. It has polar heads on this side, non-polar tails this side. Polar heads are hydrophilic, which means they're soluble in water. This is non-polar tails, they are hydrophobic, they are afraid of water, they are insoluble in water. So how these phospholipids, they're going to align themselves so that they can escape the exposure to the water, because this side of the phospholipid can face water, this side of the lipid cannot face water. So in this situation, these phospholipids, they align themselves in the form of a bilayer. So the phospholipids, they align in a way that these non-polar tails, they face each other, and these polar heads, they are on the outside. So non-polar tails, they face each other. So when they face each other, they can actually prevent the exposure of water. And polar heads, because they are hydrophilic in nature, they can face the water. So when these phospholipids, they form the plasma membrane, and this plasma membrane separates the cell from its environment, and there is water on both sides, inside as well as outside. These phospholipids, they align themselves in the form of the bilayer. By this way, the non-polar tails, they can prevent the exposure of water, and these polar heads, they can face the water. So this is the way how the plasma membrane is formed by the phospholipids, and these phospholipids, they arrange themselves in the form of the bilayers. so this is basically showing you that this is two layers. So these are the polar heads, okay, and these are the non-polar tails. Now it all depends on what kind of a membrane it is. On the basis of that, they can have different kind of fatty acids in them. So remember that phospholipid is having two fatty acids, and instead of the third fatty acid there's a phosphate group. So these two fatty acids, they can be of various kinds. They can be saturated, they can be unsaturated. That's all what we discussed. So this is the bilayer of the phospholipids. This is the outer layer, this is the inner layer. So usually bulkier molecules tend to occur in the outer layer and small molecules tend to occur in the inner layer. Okay, so these are the bonds, non-covalent bonds. So van der Waals force or hydrophobic interactions are the ones which are basically linking all these phospholipids to each other, as well as linking these phospholipids to the proteins also, which we're going to discuss soon. Okay, so the arrangement of hydrocarbon interior of the bilayer can be ordered and rigid, or it can be disordered and the fluid. So this membrane actually maintains the fluid nature, but now depending on the kind of fatty acids they have, on the basis of that they can be rigid or they can be more fluid. So if the phospholipids, they are made of saturated fatty acids. Remember that the saturated fatty acids, they have straight structure because of the single bonds. So the linear arrangement, so if the in the bilayer they are saturated fatty acids, linear arrangement of hydrocarbon chains leads to rigidity because they are all straight chains. And if the phospholipids, they have unsaturated fatty acids in them, then what is going to happen? They will have the bend or the kink in their structures, and because of that they can lead to greater fluidity. So this is just showing the same thing. This is your fatty acid, saturated fatty acid. This is a polar head, and this is the non-polar tail. So this is the unsaturated. You can see there's a bend in the structure. Now cholesterol is a steroid which is present in the membrane of animal cells, and we just saw the structure of the cholesterol. Okay, so cholesterol is actually present in between the phospholipid bilayer, and it actually it's a kind of a rigid structure, and it gives the rigidity to the membrane. Okay, so presence of cholesterol can enhance order and rigidity. Now the animal cells are less fluid because animal cells, they have cholesterol in them. Okay, they're less fluid and more rigid than the plant membranes, and plant membranes, they have a higher percentage of unsaturated fatty acids in them as compared to the animal membranes. Okay, so this is basically just showing us that the membrane, which is made of your phospholipids, if we increase the temperature, it can acquire more fluid, or the more ordered state. Okay, it can become more fluid. Okay, so basically the plasma membrane needs to maintain its fluid nature. Okay, if it becomes a gel like this, it's not going to function because it becomes solid. Then the transport will stop. If it becomes very disordered, it's again not going to function because it's going to be not fluid. So the fluid nature of the membrane is important. So with the increase in the temperature, the membrane can become more fluid and it can lose its functionality. Okay, so this slide basically talks about some of the terms. It talks about the transition temperature. So transition temperature is that temperature in which something solid can change into liquid. That's the transition temperature. But these membranes, they are maintained in the fluid form, but they should not become very fluid, and they should not even become very solid. Okay, so in both situations they can lose their functionality. So in the animal cell membranes, there is a cholesterol, and cholesterol is present in between the phospholipids. If like in the summers, if the temperature increases, this membrane can become very fluid and it can stop functioning. But because of the presence of this cholesterol in between these phospholipids, which is present in between, it is going to stop that disordered movement of these phospholipids, and it will prevent that situation. That's the role of cholesterol, which is present in phospholipids. And the plants, they have a higher concentration of unsaturated fatty acids in them, and they have like a kink in their structure, and that is also going to maintain the proper fluid nature of the membrane. Okay, so they will not let because if there are more saturated fatty acids in the membrane, so on the other hand, if the situation becomes if the temperature gets very cold, if the membrane has more saturated fatty acids, because they have state structure, they can all pack on each other and the membrane can become a gel like this. But in the plant cells, because they don't have the cholesterol in them, they have more unsaturated fatty acids in them. Because unsaturated fatty acids, they have more bends in them, and as a result of that, they will prevent the uh, packing of the fatty acids over each other, and that membrane which has unsaturated fatty acids in them, that can maintain fluid nature in these situations. Now, the proteins which are present in the membranes, they can be integral proteins. Integral proteins are those proteins which are present, which actually are part of the phospholipid. Peripheral proteins are those proteins which are actually not the part of the phospholipid bilayer. They are present actually outside the phospholipid bilayer. They're loosely attached to the membrane, and they can be attached by polar interactions or electrostatic interactions. And it's very easy to remove the peripheral proteins. Just change some salt conditions, you can easily remove them because they are actually not embedded in the phospholipid bilayer. But on the other hand, the proteins like integral proteins, they are actually embedded in the phospholipid bilayer. They are part of the phospholipid bilayer, and it is difficult to remove these kind of proteins. To remove these integral proteins, you need some detergents and all that. So the example of peripheral proteins is heterotrimeric G proteins, and the example of integral proteins is rhodopsin. So this is rhodopsin. So you can see that this is the phospholipid bilayer. This protein rhodopsin is actually part of the phospholipid bilayer, so it's embedded in the membrane. And on the other hand, this is a heterotrimeric G protein. This is a heterotrimeric G protein. It has three different subunits: alpha, beta, and gamma. This is actually not the part of the phospholipid bilayer. It's outside the phospholipid bilayer. So this is a peripheral protein, and this is a integral membrane protein. And there are some proteins which are actually anchored to some of the lipids which are present in the membrane. So these are called lipid anchored proteins. So these are the proteins. This is one protein, this is second protein. So these are the lipids which are present in the membrane. So this is like a myristic acid, this is palmitic acid. So these are the lipids which are present in the membrane, and these proteins are covalently anchored to these lipids. So here is one protein. So this protein is covalently linked to this lipid. Okay, so this is another protein. So this is this protein, this is the protein. This is getting covalently attached to this lipid by its amino group, and this is a protein which is getting attached to this lipid by the sulfur which is present in the cysteines. You know the amino acid called cysteine. So this is a third category of the proteins which are actually called lipid anchored proteins. So they are connected to the membrane by these lipid anchors. So this is all what I have already explained. So what are the functions of these membrane proteins? They can act as transport proteins. So through these transport proteins, various things. So these are some of the transport proteins. So these are the membrane proteins. Some of the proteins, they can act as transport proteins, which means they can help in the movement of things in and out. And some of the proteins, which are membrane proteins, they can act as receptors, which means they receive the signals from the outside the environment, and they can lead various things to happen inside the cell. Various events can take place inside the cells. um, which include basically um, the cell proliferation or the activation of some enzymes. So these are some of the important functions of the membrane proteins. Some of the membrane proteins can also act as enzymes. So I have already discussed what the fluid mosaic model is. So fluid mosaic model, we have already discussed. This is the structure of the plasma membrane. So that's all in this lecture. I hope everything is clear. If something is not clear, you can let me know.